Good morning, grade 8. We are now down to the examples, the one that I had promised. This is the continuation of our discussion for monohybrid and dihybrid. Okay. But let me review first the previous activity that we have. Okay. Activity number 3, which is monohybrid and dihybrid. So only one attempt as of the moment. Okay. It's from Hermoso, Carolina. Okay. Don't worry, I'll be opening this for the last time. Okay. Please inform your classmates that I am opening activity number 3. Okay. For the last time, it's monohybrid and dihybrid. We will have the thorough discussion today. That is the main reason why. Okay. By the way, how is your learning resource material? Your learning resource material? How is it? Okay, I already given you input to the learning resource material. Let's have the discussion about monohybrid and dihybrid process. Okay, let's have first these slides. I think I have it here. Again, if we're going to talk about monohybrid cross, it is a cross between two organisms with different variations, meaning to say that it carries different genes at one genetic chromosome of interest. So we'll be focusing on one chromosome of interest. Okay, let's have this one example. Let's use the whiteboard. Okay, like for instance, we will have a cross between, okay, a cross between let's have this allele first. Color of the eyes. Okay. Color of the eyes, or shall we say this one? Let's have, wait. Okay. Whiteboard. Let's have this one. T is for tall. Okay. This is the height of an individual. Small t is for short. Okay. Let's have a cross between a tall male to a homozygous tall. Homo means one because we are actually looking at 2. This is dominant and this is recessive. Okay. Dominant allele tall and recessive allele short. Okay. Let's take a look at, okay, a homozygous tall individual being crossed to a heterozygous tall female. So we have male homozygous tall and female heterozygous tall. Let's have this one. Okay. What could be the possible offspring or result? Okay, your answer should be in the Punnett Square. Okay. You just explain the variations happening there. Okay. This is usually for male. Okay. This cross is for male and this is for female. Okay. This is male and this is female here. Okay. For male, we have tall. Okay. Homozygous tall. And for female, we have heterozygous tall. Okay. What could be the possible offspring? Okay. So male has given a dominant allele tall, and the female has given dominant allele tall. Okay, as you can see, we have heterozygous tall. Okay, male had contributed dominant allele tall, and the other female has contributed dominant allele tall. So we have, again, a heterozygous tall. How about on the next Punnett square, or in the next box, we have male contributed, okay, wait. A male contributed a dominant allele tall, and the female contributed a recessive allele short. Okay, and the next or the last square, we have male contributed a dominant allele tall, and the female contributed a recessive allele short. So we have, as you can see, we have two heterozygous tall, homozygous tall rather. So the genotypic ratio, so you see genotypic or genotypes, is the genes, okay? It could be homozygous or heterozygous because we're looking at the gene. What could be the gene inside of that particular organism? So, as you can see there, okay, the genotypic ratio there is 2 homozygous tall. Homo means 1. Is 2 heterozygous because it carries what? It carries a gene for short. Okay, heterozygous tall. Okay, or 50% homozygous tall and 50% heterozygous tall. Or 50-50. Okay, how about in a phenotypic ratio? We're going to look at it in phenotypes. Phenotypic ratio means that these are all observable behavior or physical attributes, or the one that you can actually observe. Okay, as you can observe, even though heterozygous tall carries short, okay, since tall here is dominant, you can actually observe that these are all tall. Okay, they are all tall, regardless of the genes that they are actually contained. They're all tall. So this is 100% tall, or all tall, or 100%. It is 100% tall. Okay, let's have another example. How about a gene for a straight hair? Let's have a gene for straight hair, which emphasizes straight, capital S, straight, and small letter S, curly hair. Okay, so straight is dominant over curly. So let's have a cross between a heterozygous straight, okay, heterozygous straight, because it carries a gene for curly male, to a heterozygous straight female, because it carries again a recessive allele curly on its genes or genome. Okay, let's have the cross. You create a Punnett square. Okay, you create a Punnett square. Okay, male is heterozygous straight, while female the same, heterozygous straight. Okay, what could be the possible offspring? So we have the male contributed a dominant allele straight, and the female contributed dominant allele straight. While in the second box, you can actually see that the female contributed a dominant allele straight, but the male contributed a recessive allele curly. In the lower box, in the third quadrant, you can actually see that the male contributed a dominant allele straight, and the female contributed a recessive allele curly. Here in the fourth quadrant, you can actually observe that the male contributed a recessive allele curly, and the male or the female contributed the recessive allele curly. This is the main reason why this recessive allele can only be in the progeny if it is paired with another recessive allele. Okay? You can actually observe this one if it is paired with another recessive allele. So let's have the genotypic ratio. Let's check on their genes, okay, and the progeny. In the cross between a heterozygous male straight to a heterozygous female straight pair. Okay, let's take note of this. We have one homozygous straight is two. We have two heterozygous straight okay is to this one, okay, one curly, okay. That's the genotypic ratio. We're looking at their genes. So if you're going to look at their genes, it actually what? It actually resembles the cross between heterozygous male to heterozygous female straight is in their genes, the progeny, we can actually observe one homozygous straight is to two heterozygous straight is to one curly. So we can actually put it in this way, 25% heterozygous straight is to 50%, okay? 25% homozygous straight is to 50% heterozygous straight is to 25%, okay? Curly. So it is 25% is to 50 is to 25, or 1 is to 2 is to 1, or 1 is to 2 is to 1. 1 homozygous trait is to 2 heterozygous trait is to 1 curly. That's in the genotypic ratio. How about if you're going to look at the phenotypes or phenotypic ratio? Phenotypic ratio in the sense that if you're going to look at phenotypes or phenotypic ratio, you are actually talking about their appearance, observable traits, observable behaviors. What else? Regardless of what genetic characteristic did they have, what is inside their genes. Okay, we need to look at the physical attributes that they have. Okay, for phenotypes, as you can see here, this is straight, 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 and curly. So we have 3 straight, is to 1 curly. 3 straight is to 1 curly, or 75% straight is to 25% curly. Okay? Did you understand now the concept of homozygous or the monohybrid? Did you understand now the concept of monohybrids? Okay. Let's move now to dihybrid. Okay? If you're going to look at dihybrid, okay? Dihybrid is a cross between two different lines or genes. We're looking at different genes, okay, that differ in two observed traits or observable traits. Okay, it is a mixture. We are actually a mixture of different genes. Okay, now dihybrid, we are now looking at the two genetic point of interests. So let's have this one first. Okay. What if we'll have two genetic point of interest? Like for example, tall. Okay, let's mix tall and straight hair. Okay, we have tall, short. Okay, capital letter S is okay. Okay. So we have straight hair and curly. So we have two different sets of genes, okay, two different alleles, two different points of interest. So what if we will have a cross between a heterozygous tall with homozygous straight hair male to a heterozygous tall female with curly hair. Okay. Sure, how can we make this cross? What should be done first? Okay. In crossing or in order for us to have a cross of the hybrid, you need to cross it first. So you need to cross this over by itself and cross it over by itself in order for you to get some genes of interest. So we have this, you need to cross it over in order for you to have your variables. Okay. We are now having our variables here. Sir, these are different set of genes. We have two genes here. Yes. The purpose why do we need to create this one is for us to what? To have the variables inside that protein, inside that genes. Okay? Inside that genetic point of interest. So this is in male. Let's start first with male. Cross it over by itself. So we have heterozygous tall and homozygous straight. So what could be the possible result? We have TS, okay, and TS. Always start from the first genes that you started here. Okay, we started here, as you can observe, we started from, this is the genes for height. We started from the genes of height, correct? Am I correct? And the second gene is hair, correct? So you started from height, and next is hair. Okay, same here. You start from the height, which is tall. Okay, and hair. Same as here. You start from the height and hair. So these are now the variables under the male. Here we have CS, okay, small letter T and capital letter S, all capital letter T and S, okay, and small letter T, capital letter S. Okay, how about the female? Are you still following? Are you still with me? Are you still with me? Okay. You need to listen very carefully because this might be tricky in your part. You need to ask questions. Okay. Now in the female, okay, let's cross it over by themselves to have the variables of this genetic interest. Again, heterozygous tall. Okay. And the female, heterozygous tall, and the second gene is what? Is curly. Okay. This is curly. So we have, let's start again from the genes of height, okay, to the hair, genes of height to the hair, okay, genes of height to the hair. Okay, so we have now the variables TS, okay, small letter T and S, T and S, and small letter T and S. So these are the variables. We already extracted the variables of these genetic interests. Okay, what should be done next? Sure, what should be done next? Okay, the next thing to do is to cross them over. This is now the exact time that you cross the variables over. Okay, again, let's create a Punnett square. We already created a Punnett square. Okay, diba we agreed upon that this is male here and this is female here. Okay, so for the male we have TS, okay, CS, all capital TS, small letter T and capital S. For the female we have small letter T and capital letter T and S, all small letter T and S, capital letter T and S, okay, and the last one is all small letter T and small letter S. Okay, we already inputted the variables of the said traits. Let's have now the cross. Okay. You check on the cross, we have, you start from height, okay? Height, so T. You check on the T here and the T here, look at, we have homozygous tall to a heterozygous straight. Okay, we have homozygous tall, heterozygous straight. How about here? By the way, in writing genetic interests, okay, you need to start from the dominant allele. Okay, that is why we're writing capital letter here. But in the case of your variables, you need to write what is actually the first gene that is being discussed, the first gene that is actually in your given. Okay, because we started with height, you need to start this with the height and followed by the second gene, which is the hair. Okay, let's continue. We have dominant tall with short, okay, and dominant straight with curly. Okay, this is heterozygous tall and heterozygous straight, homozygous tall and curly, homozygous tall curly. We have also heterozygous tall with, this is wait. Okay, again. This is capital letters, by the way. Okay. Homozygous tall with, okay, capital letter S and small letter S. Heterozygous straight. Okay. How about in this one? We have homozygous tall. Okay. To a homozygous straight. How about in the next quadrant? We have heterozygous tall to a heterozygous straight. Okay, remember that these are capital letters. So these are dominant alleles. Okay, we have short, okay, short and heterozygous straight. Okay, heterozygous tall with heterozygous curly. Okay, short with heterozygous curly. Next is homozygous tall. Look at this one. It's all capital letter T and one capital letter S to a small capital letter to a small s. So this is homozygous tall to heterozygous straight. Okay. Again, heterozygous tall because we have capital letter T and small letter t here. Capital letter S to small letter s. So we have heterozygous straight. Homozygous tall to heterozygous straight. Short. No. Capital letter T to small letter t. This is heterozygous tall to a heterozygous straight. Okay. How about this one? Heterozygous tall. Okay. To heterozygous straight. Okay. So small letter t and small letter t. So therefore, it is short. Is to heterozygous straight. So we have a short organism with heterozygous straight hair. Okay. Heterozygous tall. Okay. To heterozygous straight hair. Are you still following? And the last quadrant, we have short. Okay. All small capital, all small t and S. Capital letter S and small letter s. So we have short and with heterozygous straight hair. So there you go. These are the possible progeny. So how can we get the genotypic first? How can you get the genotypic ratio here? Genotypic ratio. Okay. So we have 1 homozygous tall with heterozygous straight hair. 1, 2, 2, 3, 4. Okay, we have 4 homozygous tall with heterozygous straight, because it carries again a gene for curly. Okay, is 2. Let's check on the second. We have heterozygous tall and heterozygous straight. So let's look at the heterozygous tall, 1, and heterozygous straight, 2. Okay, 3, 4, 5, 6, 7. So we have 7. Again, 1, 2, 3, 4, 5, 6, 7. It's 8. Again,